Hi, everyone. For this lecture, we're going to talk about pathogens and epidemiology in general, or the science of studying infectious disease, and how infectious disease moves around and transmits from one person to another. So, as I mentioned earlier, just now, epidemiology, when we talk about epidemiology, we're talking about the study of infectious disease, not necessarily disease that is non communicable, things that can't jump from one person to another, but specifically things that are infectious, that you can contract from one person to another, or from one source to another, from your environment to you. And this as a field of study, has developed pretty substantially over a relatively short period of time. Like our understanding of epidemiology is shockingly young. This is something that has developed especially since like the mid to late 1800s, really because of the onset of our modern understanding of cell theory and germ theory. And there's a lot of people that we can thank for this. Louis Pasteur is probably one of the most influential people who contributed during this time. He especially is responsible for not just overturning spontaneous generation as this idea of the time, but also recognizing and being the first person to really put forward in clear terms germ theory, the germ theory of disease. Robert Koch was also really influential during this time. These two, by the way, hated each other's guts, and it was that hatred that really pushed their research forward, because they always wanted to outdo each other. So it should come as no surprise then that Robert Koch was very involved in germ theory as well, and was he did a lot for especially looking at the causative relationship between specific microbes and the diseases they can cause. And so that's what a lot of his work is related to. It's really important to note that before these people, before some of their predecessors, the prevailing theory at the time was miasma theory. It was this idea that disease was caused instead of by a microorganism, but by a bad smell. Right, that you could have bad air, and exposure to bad air could cause disease. So it was, it was very much an issue of people finding correlation, but not understanding causation. Understanding that, yeah, people did tend to get sick if they hung around really, really stinky places, things that smelled fetid and rotten and things like that. And we understood that when people did get sick, bad smells often came with that as well. But people didn't really understand at the time that it wasn't the bad smell that was causing that; it was the microbes that were living in the environment, living in you or that patient or whatever, that was helping you produce that bad smell or causing that bad smell. And it was people like Pasteur and Koch that helped kind of sort out reality from falsity like this. So, as we explore epidemiology, I really want to identify a few important terms. Okay, so first, infectivity. Infectivity is the ability or likelihood for a pathogen to infect another host after exposure. So, as we go through life, inevitably we get exposed to lots of different things, diseases out there, but we don't get disease every single time that we encounter it. We don't get sick every single time that we encounter disease. And so diseases have, or pathogens specifically, have a quality of infectivity that influences how likely it is that after exposure we get infected by that pathogen. Pathogenicity, however, is the ability of that pathogen to cause disease in those who are infected. It is very possible to be infected with a pathogen but not have it lead to disease. For example, if your immune system encounters it quickly and deals with it before it can manifest any real symptoms. That happens to us all the time. We don't even know it. So this is important as well, right? Pathogenicity, and then virulence as a separate measure. This is typically a measure of the disease severity that can be caused by this. This is something that gets argued a little bit here and there. Some people have slightly different definitions of virulence, but generally speaking, virulence is the ability of a pathogen to cause severe damage in those who do get infected. And so really it's a measure of that health impact for that host, where if a disease is more virulent, it is more damaging to a patient that has it. Now, I want to specifically say that disease epidemiology, this is a complicated subject. We're not going to get into everything here by any means. Okay, you'd have to take a dedicated class for that, and I'm sure you will at some point or another. But we're going to go over some important aspects here, talk about a few different things. But the important part is that this is a multi-dimensional problem. Okay, that we have to, when we start thinking about dealing with preventing infectious disease and understanding it, treating it, right? We have to understand the many different dimensions of that this infection. Not just what causes it, right? Of course, what causes it is very important, but also who is vulnerable, right? Who is actually capable of contracting these diseases? Who is most at risk? Where does the disease hide? Where can we find the disease hanging out when it isn't necessarily infecting a host like us, like you? Where do we find it? Do we find it in other populations of people? Do we find it in other organisms like animals? Do we find it in the soil? Where does it exist? That it could be encountered again. How does it enter our body, right? And in what method does it do that? How does that method influence its aspects of pathogenicity, its aspects of virulence? What about its portal of exit, right? When it leaves our body, how does it leave to infect other people or other organisms in general? Tied closely into that is the means of transmission, right? How does this disease actually get around? So this is, of course, a multidimensional issue, and we're going to explore some of these things here, but certainly not all of them in great detail. When we acquire an infection, right, an acute infection. Of course, last time we talked about different kinds of infections. You could have a primary infection that disappears and comes back, at a later time. Different things like that. We're going to talk mainly about acute infections here moving forward. So, just singular primary infections. You contract it, you fight it, and then it is gone, right? Now this acute infection typically has a pretty typical pattern of impact, right? Where we have initial point of exposure. We start with a very limited quantity of this pathogen that we've encountered here, okay? And then initially you have to have this pathogen incubate. This is the incubation period where the bacterium or the virus or the fungus, whatever it is we're dealing with, is growing. Specifically, it's reproducing, right? It is starting its process of infection, and so that ramps up, and then we get the prodromal period. This is more of like a vague sense of sickness, like, oh, I'm feeling a little under the weather, but I can't really put my finger on it. And then later you get the illness stage. This is the more severe stage where the pathogen really ramps up. It is becoming very successful at this time in this new environment, and our body is mounting a response at this point. But in this process, we are at the point where we are most sick. Okay, this is where we're getting the greatest symptoms. The greatest health impacts usually happen during this time. Okay. Then, after our body has mounted a sufficient response, where it has really escalated and now the pathogen is on its back foot, we're in the declining period where the number of microorganisms is slipping, it's declining, it's reducing, and then eventually we get the convalescence stage. This is where we no longer are experiencing symptoms, okay? And this will continue to taper off till it gets to zero eventually, okay? Now, the timeline that this happens on can vary quite a lot. It's something that could happen very short periods of time, like over a few days or a week, or for some diseases, it might take much longer, like months. Diseases kind of like mononucleosis can be sort of along those lines. It takes a long time to get rid of these symptoms. And importantly, it really does vary quite a lot with the different diseases that we're talking about. Really, there's going to be a lot of variation here. Okay, a lot of variation in sort of like what is like most significant and most notable during this time. Okay, now it's important to remember when we talk about pathogens that when we describe a pathogen, we're talking about a parasite, okay? Where this is a form of parasite. Remember that all pathogens are parasites, but not all parasites are pathogens, right? A pathogen is just a parasite that causes disease. An organism that is a parasite is one that sustains itself on the resources of that living organism. Okay. Now, pathogens, of course, fit into this category as well. Even viruses, viral particles, even though they're not alive and they don't feed on anything, they still are very much sustaining themselves like a parasite would. And so we consider them to be parasites as well, right? Even though they're not alive, we still call them parasites because they're using the cell's resources or the body's resources to build more of themselves, right, at the cost of that cell. So as we explore these pathogens, we need to start asking ourselves important questions like, what is the goal of a pathogen? What are they doing, and why do they do what they do? It's not because they're some evil force in this world, although it may sometimes be easy for us to think about it like that. Ultimately, this all comes down to it just being biology. These organisms are following their basic biology to do their jobs. And so, the better we understand the biology that they're experiencing, the better we understand the organisms themselves and the diseases that they cause. So, okay, what is the deal, right? What is the deal with these pathogens? What are their goals, right? Their interests, their purpose is driven by just that base biological need to reproduce. That's pretty much it, right? So this is ultimately just, everything they do, all the different activities that they do, all the harm they cause is just in the furtherance of their need to reproduce. So their method to do this, right? Their specific method is just happens to be problematic for organisms that they parasitize, right? We get infected with these organisms, they steal resources from us, and they use our bodies to reproduce and make more of themselves. That's just their specific strategy. And so their unique strategy has a unique life cycle that goes along with it, right? That life cycle looks like this. First, the organism has to find a viable host. Not every host is going to be viable. They need to sort out the viable from the non-viable and hopefully infect the viable ones. Once they achieve this, they enter into the host's body and they find a home amongst its cells. Okay. Once it's there, its next job is going to be gathering and gaining access to the various resources of the body. Okay. So it does a cell no good, it does a pathogen no good to make it into a body without being able to utilize those resources. So that's the next step, is figuring out how to do that. From there, it's going to be all the while evading detection and trying to avoid being killed and removed by the host's immune system. And all the while, it's going to be focused on reproduction, just reproduction. Okay, and after they have reproduced a lot, they're going to be then trying to spread their offspring outside of the host to restart the cycle with a different host, one that is hopefully not infected with it or something else, and basically fertile ground to establish and grow in. So let's keep on going here. Pathogens, their life cycle there are various jobs that it has to do, and the evolution of these pathogens is in furtherance of that biological function. So a good pathogen will have evolved to be effective at these different strategies, and so it will have a number of traits that help it along the way to do this job. So for example, the job of, once a pathogen makes it into the body, it has to be able to find a home in there. It needs to be able to stay in there and hold on to something and oftentimes get an anchor on the location that they really want to live on. And so to do that, a lot of bacteria utilize adhesion factors. It's something that bacteria do, viruses do. A lot of pathogens will do this, and they use these specific proteins and other features to help them hold on to the surface of a host cell. Okay, the mechanism is pretty interesting because they essentially, these cells, they produce these compounds called adhesins. These are usually proteins. And the proteins sit on the outside of the cell, or in the case of a virus, the capsid or the membrane, and they have, just like a lock and key mechanism would usually find with a receptor and the thing that it's supposed to bind to. We find the same thing happen where these adhesins fit really well into receptors that are on the host cell. Okay. Now, it's important to remember that these host cells, they didn't evolve these receptors so that they could bind with and hold on to bacteria or viruses. It's far from it, right? This is the pathogen adapting to utilize a feature that the cell already has, that their host has, right? That's why they have this feature. It has nothing to do with the host. In fact, the host, if they could help it, they would prefer to not have an adapter that matches these adhesins. Okay, this is part of the sort of back and forth evolutionary cycle that these organisms are under with their hosts or with their pathogens. So if the pathogen's adhesin fits the host receptor, it binds there, it holds on, and so it prevents the pathogen from dislodging from the host that it wants to infect. Okay, but if it doesn't if it doesn't adhere, if the adhesin does not fit like a lock and key into the host receptors, you get no adhesion. Okay, and so likely you don't get infection either. So this is really important because these receptors are going to vary quite a lot with different organisms. The more distantly related different organisms are, the more likely it is that their receptors don't match. And so this is another element that contributes to what we call host specificity in a pathogen, the very specific nature of a pathogen to parasitize only specific species and oftentimes also only specific cell types within those species, right? For example, a disease that can only parasitize cells in the respiratory system of a host, but can't make its way for various reasons into the liver or the muscle tissue or the nervous tissue, right? A great example of this is forgive me, I got to say these names, Neisseria gonorrhoeae. Some of these are easier to say than others. This is the causative agent for gonorrhea, and this species is pretty interesting because it utilizes some pretty specialized adhesins to attach to very specific cells. So it comes in, it interacts with and binds to cells with these specific receptors on the urethra, the vagina, and also individual sperm cells. It can also parasitize. So it's looking for it pretty much attacks like these membranes inside of these organs and then these individual cells. And so because we find that other body cells lack the receptors to bind to the adhesins here, the infection remains pretty localized in the case of gonorrhea. It doesn't infect more broadly outside of these sexual organs and these specific cells. The cells are pretty important because it also helps the infection spread through intercourse, right? So in addition, we have various other mechanisms. We'll talk about a few of these, okay? When we talk about gaining nutrients, these pathogens have additional strategies and have additional problems to overcome. For one, the body doesn't want to part with any of its resources, so it has to take them, with force. And so one tactic that a lot of these pathogens use is they actively dissolve cell structures, body structures. They break things down, and for digestion, or to, if not to directly digest those things, but to release things that are hidden behind additional layers of protection, other kinds of nutrients, right? Like to lyse cells and break into those cells to kind of get all the goodies inside. To do this, these pathogens utilize exotoxins and endotoxins. Okay, exotoxins are a chemical that is produced by a pathogen and then secreted by it, so it's released outside of the cell, and it goes out to break down or destroy other features of the body or specific cells. And there's a lot of classifications we could give exotoxins. That you could have compounds that are cytotoxins, or we describe them as being cytotoxic, right? So a cytotoxin is a type of exotoxin that will kill host cells or directly negatively impact their functions, right? It is toxic to those host cells. Neurotoxins are specifically directly or indirectly damaging to nerves, right? The nervous system. Enterotoxins are damaging directly or indirectly to the gastrointestinal tract. Let me see. Hemolysins are toxins that are destructive for host red blood cells. So we can actually see that anytime we do a blood agar plate and we get different kinds of hemolysis. If the pathogen is going to do some hemolysis, either alpha or beta, it is going to be breaking apart some or all of the red blood cells that it encounters, or partially or completely. We can also have membrane damaging proteins and or toxins. These are they're often proteins, but they aren't necessarily. And these are toxins that will interfere with the cell's plasma membrane. So you will find that hemolysins tend to be membrane damaging toxins, for example, because they break apart those cells. On the other end of this spectrum, we have endotoxins. Endotoxins are compounds that the pathogens produce, but they keep inside their own bodies. They're not secreted. So a good example of this would be like the lipopolysaccharides that we talked about in gram-negative species. So these are specialized endotoxins that are adorned on the outside of the cell, right? On the outside of the outer membrane, right? Now, remember, it's not secreted. It's not released from the cell, so it remains an endotoxin even though it faces the outside environment. It's still affixed completely to the cell. And these lipopolysaccharides and other kinds of endotoxins, they become real problems mainly when the cells that carry them die or are digested or something like that, or even when they undergo division. When the cells divide, they can sometimes shed some of these things. And so when this happens, that can cause a toxic effect that comes from it. Maybe it's cytotoxic, maybe it's neurotoxic, etc. Now, avoiding the body's immune system is also a pretty high priority for these pathogens, because once they establish and once they start feeding, well, the body is going to take notice of this, and they're going to want to get rid of them. And the body has many different mechanisms to do this, many of which we're going to talk about in the next chapter. But there are many different strategies they're going to be using. And so because of that, the pathogens have many different strategies they have to try to avoid as well, or work around. So a lot of pathogen defenses are going to look somewhat similar. They're going to fall into a few different categories. For one of the more important jobs is going to be trying to avoid being captured by our phagocytes. Remember that our phagocytes, you've likely encountered this in previous classes by now, that phagocytes are human white blood cells, specialized white blood cells, that roam around, and they're very mobile, like this, and they are very search and destroy. Their job is to track down and swallow and ultimately digest anything that the body recognizes as foreign. Okay, these are basically the janitors or the vacuum cleaners, or however you want to think about it. Now we have phagocyte activity in a lot of different places that you might not necessarily think. They're just widely active. You will even sometimes have, virtually always, you'll have phagocyte activity trying to work to destroy, I don't know, if you have a tattoo. The foreign material that is put there in your body during a tattoo, it's inside your body, even though it is not all that deep. And you have these phagocytes that your body is sending them signals every now and again that says, hey, this thing doesn't belong here, and your body tries to come up and clean it. But in the case of your tattoo, if you have one, the ink is too large. It's too large of a molecule to be easily swallowed by these things. It's quite large, so it's not very easy for them to clean this up. That's why our tattoos stay there for quite some time. But if you were to go and get a laser, what's it called? You go to get your tattoo lasered off, right? Well, the laser is just going to break up those large molecules of ink into smaller and smaller pieces, and then your phagocytes come in and they clean that stuff up, and then it goes away. So they're very involved in this, and so your phagocytes, right? They're very involved in cleaning up anything that they register to be foreign, and so when your body recognizes there's a foreign invader there, a bacterium, a virus, whatever, it's going to be tackling those too. So avoiding being captured by a phagocyte is important. You can actually see look, this in this bottom GIF right here, it almost looks like for a bit this bacterium is kind of fleeing from this phagocyte, and it probably is to some degree avoiding the phagocyte because it is mobile, or it likely is, and it does not want to be eaten. It's getting senses from its environment to tell it that a cell that it needs to avoid is nearby, and it's in this direction, so it moves away from it. Alternatively, the pathogens may be trying to avoid being digested by a phagocyte. If a bacterium is swallowed by a phagocyte but cannot be digested, it sits around there and it keeps on doing its job, and it reproduces and reproduces. Maybe it even steals from our phagocytes and things like that. This is incidentally how tuberculosis sticks around. Once you get a tuberculosis infection, your body's phagocytes actually clean it up really quickly, but it can't digest it whatsoever. They're just too tough for our phagocyte lysosomes to tackle. So they kind of stay in a sort of stasis of a sense until those cells die ultimately, because every cell, these phagocytes don't live forever. Alternatively, you have antibody activity inside of your body, and so the pathogen may be evolving mechanisms to, or may have already evolved mechanisms to avoid these antibodies binding to the cell, right? That may have specific tactics that help it avoid actually interacting with those antibodies. If an antibody binds to a pathogen, it can kind of clog it up a little bit, prevent it from interacting with its environment. But if the antibody can't recognize or doesn't have a feature on the bacterium to bind to, there's no binding. Okay. Alternatively, the pathogen may have evolved mechanisms to try to break down antibodies after they have bound to it, or just in the cell's environment. Usually in the cell's environment. I think that usually once they bind to the cell, it's kind of hard to deal with them. But the antibodies in the environment are more fair game, and so there are some pathogens that will tackle that. That's their main goal. Okay, now it's about time that we switch gears. We're going to talk now instead about the adaptations of these pathogens for their infection. We're actually going to talk about more of the epidemiology, the spread, transmission of these diseases, and I want to actually explore the spread of disease in a little bit different way, a subject that we don't usually talk about in this class because it's not a human that is getting this disease. I want to talk about chestnut blight as a great example to talk about the spread of disease. Okay, so historically, North American forests, particularly on the eastern side of the U.S., and especially in the Appalachian Mountains, the mountain range down here, all the way up here, we had these forests used to be very dominated by a particular tree called the American chestnut. Now, this species was so dominant that there were an estimated many billions of trees that populated this area here. And it was the main constituent of these forests. Like how today we might go outside and find like an oak and maple forest or maybe a pine forest where these forests are mostly dominated by, one or a few species. This was very much the case for North American forests at the time. It was mostly American chestnut, even though today it is not that case anymore. And the reason why it is not the case anymore is twofold. First, of course, in this early time, I'm talking about like early colonization of North America by Europeans, and maybe the next like well, I mean, all the way until now, right? Really, what we're talking about. But before about 1900, these forests were dominated by these trees, and of course, after Europeans arrived in North America, logging commenced to an extreme degree, and so a lot of these forests were in places clear cut. But believe it or not, that wasn't actually the greatest threat to the American chestnut. Instead, it was a specific disease called chestnut blight. This is a fungal infection that was endemic, or like native to East Asia, and it was introduced initially through imported Japanese chestnut trees. So we brought chestnut trees from Japan, specific Japanese chestnut species, to actually New York. Actually, it's the New York Botanical Garden who is sort of responsible for this in a weird way, because that is where these trees ended up. They were brought over to NYBG and planted there, but those trees, well, they carried chestnut blight with them, and so the first cases emerged there in New York City, right? The disease itself, when it manifests, causes a lot of these cankers that emerge from the tree itself. This is part of the symptoms of this disease. So you get this like pock marking of these fungal cankers that erupt from the bark, causes a lot of damage to the tree, persists over months and months. And it has a very, very high rate of mortality for infected trees. If a tree is infected with chestnut blight, it's probably going to die. Now, to talk about specific terms, okay, this first instance of a few cases that emerge in New York City here, this is the disease outbreak. This is the specific location of that outbreak, and it is the outbreak itself, right? An outbreak is just the occurrence of a disease in a significantly greater abundance than what is expected in the region, right? So, let's talk about that in a different form. A situation where you have a new disease, right? Either an emerging disease that was that was novel to the specific region or a specific population, or a re-emerging disease, i.e., a familiar disease that was already present in that region or already existed in that population, but it was at low numbers or maybe gone, and now it returns, right? Well, that number, that significantly greater abundance that we're talking about up here in this definition, that number might be 0, right? So, of course, or 1, I should say. So the occurrence of a disease in a significantly higher abundance than what is expected in the region. That number that is expected might be 0 because it's not found here, or maybe it's gone. And then a new case may very well be considered an outbreak. But often we think about outbreaks being multiple cases, especially if one person contracts or one organism contracts the disease from another organism, and there seems to be a chain of events. Okay? So that is really important to think about here. Now, an outbreak does not necessarily mean that there's going to be a long-term crisis. Okay? An outbreak ultimately is just an unusually large number of cases of a specific disease. Again, that number might be 1 or 2 or 3 or maybe 50. It really depends on the context. An outbreak of chestnut blight in this case, because it was new to this region, could be considered 0 or 1, I should say, 1 over 0. So, because it's just not expected in this region. But if you're talking about a more familiar disease, like an outbreak of, influenza, right? Well, influenza is pretty familiar to us. We deal with it, every year. essentially, sometimes multiple times a year, and so, or throughout the year, I should say. And so, an outbreak is not necessarily 0 or 1. It's something more significant. It's hundreds or thousands perhaps of cases. Again, context dependent, right? But if an outbreak continues and is perpetuated, and especially if it continues to spread and grow, then that outbreak can turn into an epidemic, right? An epidemic is the rapid spread of a disease in a population and a region, okay? So, particularly in a short time is important here, that we're looking at the rapid spread of a disease, that it continues to jump and expand and grow, across a larger and larger group of people, or cases, and also a larger and larger geographic area, right? So this is an epidemic, right? Is where you get this rapid spread. So modern examples of epidemics, right? In recent time, okay. Of course, Zika, swine flu, etc., different things like that. They were epidemics in the past. These are specifically ones that I picked that ended, right? Now we have epidemics that have not ended as of the recording of this video, like, for example, COVID. COVID is very much still an epidemic. It is something that has just simply become so incredibly widespread that it continues to perpetuate itself. And it will rise and fall and continue to change over time moving forward, okay? But it certainly was an epidemic and still is an epidemic in this case. And we'll talk about, by the way, what this means when you have an epidemic that sticks around and becomes global in this case, right? So we'll talk about that in a minute. But when a disease when it perpetuates and persists for a long time in a population, it can eventually be considered to be endemic. Okay, endemic. So this is a condition that has two meanings. Okay. In biology, generally, I even used it earlier, it just means native to a particular region, okay? That this particular species is from this area. But in epidemiology, it's a little different. What it really means in epidemiology is that it is a disease that is constantly and consistently present in a particular region, and now occurs just kind of naturally in a subset of the population. It's always going to be here. It just is. It circulates and circulates and circulates, okay? So this is important to know, and it's important to distinguish between these things, right? Because endemic diseases can still have outbreaks as well, and even still become epidemics. That you can have diseases that are, they have occurred here for so long that they're just local, they're endemic, but they can have sudden explosions of occurrences. Or they can suddenly become a problem again that really rampages through the population, right? So that is important to remember. Just because a species or a type of disease becomes endemic does not mean that that it's no longer going to be a problem, right? Examples of diseases that are endemic but still can have outbreaks, and we still are afraid of there being epidemics. Things like measles. Measles has historically been in the U.S. for ages. It has followed us everywhere we've gone. It is a kind of a universal problem, and measles has gotten so much better over time because of vaccines. Exclusively because of vaccines, by the way. Vaccines are the major only thing that is contributing to the the low rate, extremely low rate of measles in the U.S. and in most parts of the world as well. But that does not stop measles from having outbreaks every now and again, particularly in unvaccinated populations. So there are cases every now and again, you hear about them in populations of communities that are notoriously undervaccinated. So these outbreaks occur, and thankfully, until this point, we have not had a measles epidemic again in this country. But we still have outbreaks now and again that are certainly very concerning, and definitely becoming more and more of a concern as questioning of vaccine science becomes more prominent in popular culture. Now, a pandemic. A pandemic is a disease that will infect organisms across a huge range, right? Very expansive geographic range, not necessarily globally, but across multiple regions, okay? But oftentimes we do find that pandemics are global, okay? Now, a pandemic is kind of an opaque term, which is very difficult, kind of notoriously difficult to solidly define. The above definition is a good one, but it is not the best one. Okay, and what qualifies as a pandemic is sometimes debated. All right, and this is pretty noteworthy because pandemics. We want to understand pandemics as well as we can because they are the greatest global problem, or one of the greatest global problems that we may face. And so there are several diseases that are global pandemics. HIV and COVID-19 are good examples of now global pandemics. Again, these are diseases that were at one point outbreaks. They are now, and they since became epidemics and are now considered pandemics. They can still have local outbreaks. I want to specify that as well. So just because something is a pandemic does not also mean that it cannot have localized outbreaks that can go on to become greater problems. Okay, so another thing that we're going to talk about here is disease transmission. All right, disease transmission is the act of being passed from one host to another, from an infected host to an uninfected host, to eventually make them sick and perpetuate the disease. Right now, transmission is something that we can group some different ways. These groupings are not mutually exclusive; they can overlap, but they're useful for us in how we talk about transmission. Okay, so I want you to keep these in mind. Three major categories: we have contact transmission, vehicle transmission, and vector transmission. Now, within contact transmission, we can have direct or indirect contact, right? So, direct contact is when you have very direct contact with an infected organism, and so that direct contact facilitates transmission of the disease to you or to some other patient. Indirect contact is, again, indirect contact with an infected organism, specifically their environment, the things that they touch or interact with, the space that they were existing in before, the fluids that they produce, different things like that. Okay. Kind of in the same category as this is droplet transmission, and this is kind of interesting because you have the capacity to cough and sneeze and things like that, and in doing so you unintentionally produce droplets of saliva, mucus, things like that, and those small droplets they are capable of holding infectious material, And they're also capable of hanging out in the air for some solid amount of time. So this is kind of along the lines of indirect contact, because coming into contact with droplets after the fact can definitely spread the disease, spread a disease that is transmitted that way. But it's not direct contact either. So it kind of falls under that window. Okay, but consider it independently for now. Vehicle transmission refers to the method that the pathogen uses for transit. Okay, so they can be airborne, where they hang out in the air. They can be waterborne, hanging out in the water. Bloodborne, where they exist in blood. They could be foodborne, where they exist, of course, in food, or they use food or drink as modes of transmission, right? Vectors. Vector transmission is rather interesting as well. Okay, so we actually have two different types of vectors that we want to talk about here: biological vectors versus mechanical vectors. Okay, a biological vector is typically a biting carrier species, some kind of organism that carries the pathogen inside their body, keyword inside their body, and then transmits it by biting or spitting on or making contact with, in that way, some additional host. Okay, and the exchange of fluids like that is what facilitates the movement of the transmission of that disease. Mechanical vectors are a little different. They are carrier species that hold the disease on their body, on the outside of their body, either in their skin or on their skin, or hair, different things like that. Okay, so there's lots of organisms, like for example, a fly. If a fly was carrying a disease on its body and it landed on your food, spat up on your food to do digestion and things like that, that can move the disease that's on its body onto your food, and then you eat that food to bring that disease into your body, to bring that pathogen into your body, right? And don't forget, there is overlap between these classifications. For example, a bloodborne transmission is also indirect contact, right? That's indirect contact with an infected organism. Encountering droplets in the air is both vehicular and it's vehicle transmission and contact transmission, right? These are just broad categories to think about these things, and you will find overlap between them. So, let's talk about some of these modes of transmission. Some important terms. So, direct contact can be pretty. This is very. We're talking about direct contact. It's kind of hard, I think, to misunderstand this, right? Direct contact involves direct interaction with infected material from that host that they have on them, right? But it's so this involves that sexual contact, skin to fluid contact, or skin to skin contact, different things like that. But it can also involve, and this is where things get a little muddy, touching contaminated surfaces, right? Or eating contaminated food, different things like that. Okay, along these lines, and this is where it blends into indirect contact a little bit. Fomites are a very important concept I want you to understand, right? Fomites are a material that is likely to carry a pathogen, okay? So they carry an infection with it. So clothing, tools, structures and surfaces, different things like that. If, for example, you're treating a patient and you don't change gloves between that patient, well, gloves certainly can be fomites. They're meant, though, to be disposable. So that when you're done treating this patient, you take off the gloves, throw them away, put on a new pair of gloves. No longer is that glove, contaminated, right? But that glove still is a fomite in that it is a material that is likely to carry that infection, right? Again, if kind of treated carelessly. A good example of this in real life outside of medicine would be bats and white nose syndrome. Okay, white nose syndrome. This is a fairly recent fungal epidemic in the United States and Canada. This is impacting American bats, and it's very deadly. We've seen about 90% declines in many bat species. So bats have become much, much, much less common in the U.S. because of this. And so it's a fungal disease, another fungal disease, that spreads through, direct contact between infected and non-infected bats, right? So these bats, especially in the wintertime, are going to, buddy up together. They're going to huddle together for warmth. They're going to live close enough that the fungus can spread from one to the next. But also it can spread through the fungal spores that linger in the air or attach to surfaces. And so we've learned that humans can potentially unintentionally move these spores from one cave to another when spelunkers, people who go caving, when they don't clean their gear between caves. So this guy is crawling in all this muck and everything like that. If this is a cave that has a white nose syndrome outbreak, his clothing is fully contaminated right now, or very likely is, and so these materials, they need to be extremely cleaned. They need to be fully and completely cleaned before being moved to another cave, or else they could reintroduce or introduce spores from this fungus into this cave, whatever cave he goes to next, which could infect those bats there. Like I mentioned before, some diseases are capable of being transmitted through droplets, like water droplets, and also something we call aerosols. Okay, aerosols. So aerosols are particular water particulate that is exceptionally small, right? Both of these things, water droplets and aerosols, can linger in the air for a long time, and they may carry with them bacteria or viruses that were coughed or sneezed out during that, like in that aerosol. Okay, this is what we consider to be an airborne transmission. So the larger droplets, right, the water droplets themselves, they may only linger for a few seconds, right? They may hang in the air for a handful of seconds and then fall. But smaller aerosols can actually linger for a long time. We're talking about minutes to hours of time that potentially infected aerosol material can linger in the air. Okay, and so a lot of diseases, especially respiratory diseases, they spread using these aerosols, especially. A lot of viruses really like the aerosols because the viruses are even smaller, so they don't weigh much at all. It helps them, they can exist in these aerosols and stay airborne for quite some time, whereas a heavier bacterium may be more prone to falling, different things like that. So this, and by the way, the aerosol range is very significant. So an infected host may have, they certainly house a lot of infected material on them, but their environment around them, because they may be coughing or sneezing and spreading aerosol around, they remain very infectious in their general area. The general area around them now becomes a hazard. So we can see, pretty substantial spreading ability. This is a person doing a sneeze. That the act of sneezing is, it really propels a lot of material out, right? So this over here, this is more that thinner aerosol material. The larger particles are being thrown a bit further away, but that material is going to linger in its environment, in this environment for some time. So, different infectious diseases are going to vary in their modes of transmission, right? Where some avenues just simply aren't going to be effective strategies for them, while other ones will be. A lot of that comes down to where this infection is occurring. For example, if it's a skin infection, well, you're probably not going to be spreading a skin infection through respiratory droplets, but you will likely be spreading it through skin-to-skin contact. Or if it's a respiratory infection, you're really looking at mostly moving things with the respiratory droplets, right? Not so much through sexual contact, right? So it really varies. It varies quite a lot. Okay, and of course, when you have especially diseases that are blood or fluid borne, that becomes extra concerning for medical staff, right? Certainly medical staff, but also the populace in general, but certainly anyone who's treating somebody with these diseases, and it creates an increased importance for PPE and aseptic technique, because when you bring a patient into a hospital setting or into anywhere where they're going to be treated, and you're going to treat them for an infectious disease, well, they may very well be spreading that disease totally unconscious, unintentionally, because they're sneezing, because they're coughing, or maybe they're bleeding, or maybe they're sweating, or maybe they're throwing up. All these different things. And we have a duty to keep ourselves safe, but also to keep those that also we're treating safe. So we have to make sure that we are wearing PPE, doing our various aseptic techniques, not just to keep ourselves safe from the diseases that we are interacting with, but also from keeping it away from the patients around us, the ones that we're going to be treating. Let's talk about vectors really quickly. Okay, vectors are a way that pathogens can hitch a ride on another organism, basically. Okay, it's essentially a living organism that carries a parasite or a pathogen. Okay, and when that organism moves and interacts with a different organism, it creates a scenario where that pathogen might jump from one host to another, right? In the vector, the pathogen itself doesn't necessarily cause the same disease, right? It usually is either asymptomatic or it's very mild by comparison. And so it's essentially just an avenue of spreading. That's all that it really is. These organisms are just taxis. So this is pretty interesting because it can also provide situations where diseases can spread both from animals to animals, like from cats to cats or dogs to dogs, or from cows to cows, what have you, by being, for example, bit by a mosquito or a sandfly or a tick or something like that, and then that tick goes to bite another one of these animals. But it also creates the opportunity for diseases to move this way too into humans, or from things to move from humans into other animals. So it creates this possibility for, if you think back to our early descriptions of this, of zoonoses, right? These zoonotic events. So these vectors are really effective mechanisms of creating zoonoses, right? This could be, it can take a few different forms. It might be a new emerging disease, kind of like COVID was, where you get, a new mutation or strain that through these vectors can jump from one host to another, from one species to another, or it could be a familiar species of pathogen that just works its way between different species of hosts. A good example of that would be something like Lyme disease, right? Lyme disease is something that gets passed around through different organisms, different animals, because it's problematic for humans, of course, but we get it through bites from ticks. Okay, but the ticks are getting that from biting other organisms. So, importantly, because of the capacity for zoonoses, human pathogens, some of them, can survive in other species even when humans have fully gotten rid of them in our population. This is what we call a reservoir, right? A reservoir is just a source of a pathogen in another population. Another population might be a different population of people, or it might be a different population of some other species, right? And basically it allows the disease to be perpetuated elsewhere. So even if we get rid of it in our population, it doesn't completely eradicate the source. It doesn't eliminate the possibility that the disease can't be re-encountered, right? And so a reservoir could be a living population, could be like a live source, like for example, if I don't know, if all of Europe and Asia eliminated eliminated measles from their population, wouldn't it be nice? It'd be great. But in North America, measles had not been eliminated, right? Well, Europe and Asia could contract measles again, could re-encounter it through travels with the United States or Canada or Mexico or whatever, right? So in that way, even other populations of people could be considered reservoirs, right? But in addition, reservoirs could even be non-living sources like remnants of a living population or waste material or some other non-living source. For example, if we go back to our talk about chestnut blight, even when the tree dies, the tree that is infected with it, even when it dies and falls, it isn't necessarily the end of that pathogen. The stump and the rotting wood of that tree could continue to sustain the chestnut blight for years and years to come. So even those dead trees can serve as reservoirs for chestnut blight. This, by the way, is what those trees used to look like. We used to have massive, massive chestnut trees that were pretty crazy. Don't see these anymore. So this is what I was talking about. This is a really, really old chestnut trunk that has died. The tree has died, and now it is just the rotting wood. But if this was still infected with the fungus, it may still survive here, right? And so these neighboring trees might not be safe. When we talk about zoonoses, it's important to remember a few things. We can contract zoonoses in a few different ways, right? Zoonoses are possible through direct contact with the species itself, through the vectors of that species, as we just mentioned, or through those indirect avenues of interaction, like waste materials or bedding or burrows, dens, shed material, things like that. So some pretty prominent examples of that we can find out here. Right now, a major problem that we're experiencing in different parts of the world is a disease called raccoon roundworm. This is a parasite. It's a roundworm parasite that is very dangerous, and you can encounter it by living too close to or interacting too much with the waste material, the urine, feces of raccoons. Not all raccoons are infected with raccoon roundworm, but some of them are. And when raccoons live too close to humans and we interact with their waste materials, we can potentially contract that disease as well. This is more a problem now with other parts of the world because raccoons are now invasive in many places around the world. So raccoons have been taken to lots of different places, and like in Japan, for example. And now in those places, these populations are thriving in those environments, and they've taken with them some of their parasites. Now they're becoming a problem for humans there. Of course, common vectors might include ticks, for example. So a tick can take bites and drink blood and exchange saliva between different mammals like, I don't know, deer, mice, and humans. And in the process, it can move different diseases like Lyme disease between us and them. And then our direct and prolonged interaction with different animals like flying foxes, like these, they create scenarios where if there are diseases that exist here, we could potentially contract them ourselves, much like we did from COVID, right? COVID now, best of our understanding, seems to be caused by our close interaction with bats and probably some other organisms in wet markets, right? Wet markets in China. So, like I mentioned, a lot of vectors are biting arthropods, a lot of insects, very common here. They are parasites in their own right, of course, but they are not pathogens. They may, however, spread pathogens as vectors. Vectors are just avenues of facilitating that disease transmission, from moving it from one patient to another, okay? And this is ultimately done by fluid exchange, right? Once again, because these ticks and mosquitoes and the like will drink blood, exchange saliva at the same time. Sometimes they will even spit up blood during that process, and in this process they can return or spread fluids from their last bite into their newest bite, right? This, by the way, is a reason why you should not damage a tick while it is biting you. A lot of people think that what you should do to deal with a tick bite, you've been bit by a tick and the tick won't let go of you, is take a match and burn the back of the tick. The tick will release at that point, but in the process, it tends to spit up kind of in panic as it pulls its head out. You don't want that. There are other mechanisms to remove a tick, typically a more manual effort to remove a tick with like a pair of dedicated tweezers, grabbing the head itself, not the body, but the head. If you grab the head or the body, you're basically like squeezing a little blood bag and you can spit stuff back up into you. The careful removal is a very effective way to help prevent the spread of that disease, right? It's one thing we can do. Now, not all biting creatures are necessarily, going to be vectors, right? But there will be some non-biting creatures that will do this as well, right? That these are the mechanical vectors that by them walking around, interacting with, and coming into close contact with other uninfected organisms can potentially spread that disease from them to the new patient, right? Without ever biting, without ever exchanging saliva or anything, right? Very indirect contact. That's what I'm saying. Let's talk about ports of entry, okay? Port or portal of entry, you'll often hear it described as, I usually say port of entry, that these are the avenues by which a disease enters the body, okay? It is the route of infection, okay? Now, there are many different pathways that are possible for an infection to pass through the body, right? Lots of different mechanisms, like, for example, moving through a body cavity. For example, oral cavity, nasal cavity, our genitals, maybe one avenue, different things like that. So we may breathe something in, we may consume something, we may encounter these things through sexual contact, different things like that. passage through a vulnerable membrane is a possibility as well. We have some pretty vulnerable membranes on our body, our sinuses, our eyes, not just our eyes themselves, but also like the skin around our eyes, the conjunctiva, is sensitive and capable of being infected. Our respiratory system and so on. Even direct passage through the skin is possible. And this may look a few different ways. It may mean literally passing between cells or cracks in the skin, or possibly a physical injury that pushes pathogens past our defenses. So different ports of entry are going to be viable for different species of pathogen. Again, remember that not all diseases are going to work the exact same way, right? They're not going to have the same identical life cycle. They're not going to depend on the same routes of entry or exit, right? So this, let's see. There's a few different ways that these bacteria could get, these pathogens anyway, could get access to us. But we also have various defenses along those paths to prevent that, right? So we have in our mucous membranes, for example, we have a lot of defenses. We have mucus secretions. It's going to be very useful for us. We also have our roaming immune defense cells and also the various layers of our epithelial protection. We have our respiratory tract that is a viable path, but also that is protected. That is protected by not just our mucus secretions again. Mucus secretions, by the way, very common method of protection, but also our ciliary cells, which are actively removing material, our cough actions, and again, epithelial layers. Our gastrointestinal tract is protected not just by mucus, but also by our extreme pH of the stomach and also our epithelial layers, right? Now, methods that bypass these defenses are typically provide more success for an infecting organism, okay? An infecting pathogen. So, for example, an infected cut or burn or a poke with a needle or stepping on a nail will help bypass the skin protection, right? Anything that allows bacteria direct access to a wound or something that will push bacteria past a boundary. For example, stepping on a nail and creating a path directly through all that stuff. Anything that was on that nail is now way up there in that wound. Okay, and so here's a nice figure that kind of shows for just COVID that COVID can follow a few different paths to get into your body, but it's mainly got like 3 ports of entry essentially, right? So the ports of entry are your eyes, nose, and mouth, right? Those are the main avenues, and particularly your nose and mouth, of course, are going to be the main paths forward. Essentially, these soft membranes in here and getting into your lungs are going to be your major paths of infection, right? So this is the main avenue that you get this thing, but it can also infect a variety of other parts of our body, right? From there, the mechanism that it does this though is focused on these ports of entry. You're not going to get COVID through contact on your skin unless that skin then makes contact with this port of entry. And of course, not all pathogens can use all pathways either, right? This really depends on the specific biology of those pathogens. For example, cholera. Cholera is a waterborne disease. By necessity, it can't be transmitted through nasal droplets. it's not going to pass out that way, and it's not going to be breathed in that way either. It mainly needs to get in through our digestive tract, right? And so for that reason, again, like not all ports of entry are useful to all pathogens, right? You're going to have some ports of entry that are going to be dead ends. For example, if we have Staph aureus that lives on our body, right? And we know that Staph aureus can cause disease in specific situations. Staph cannot cause disease usually inside of our body if, for example, we ate some of it, right? If I really, I don't know, spent a lot of time like licking my fingers after I ate, I don't know, chicken wings, right? I eat chicken wings and I have staph that lives on my body, right? Well, then I lick my fingers after I eat, buffalo wings, whatever, and some of that staph could potentially get into my body that way, right? Its port of entry in that case is my mouth. I eat that, it goes all the way down into my stomach, and then, well, the staph can't survive the extreme pH of my stomach, and so it dies. That's that. That's the end of that story for staph. Now, some ports of entry for that reason are dead ends. Others are just simply more challenging, where an infection is possible. It's still possible to do that, but it's going to be moving much more slowly or be less effective during that time. And there may be, related to that, there may be less risk for that patient, making them more able to fight off that infection, perhaps. Or the patient risk might just be different. It might even be more intense under some circumstances. So it's important to note that these ports of entry and how they affect the host and also the speed or timing of infection, it can vary a lot. The example I want to talk about with that is smallpox, okay? Because I feel like we don't really talk too much about smallpox nowadays. But smallpox was a learning experience in a way for the importance of ports of entry, especially in early days. So smallpox, it's easy to forget, those infections are incredibly old. We can see evidence of smallpox infections all the way back in 3,000 years ago. Egyptian mummies bear these scars still, which is really interesting. And of course, it's likely much, much older than that. This is a long time human disease that we have thankfully dealt with. So this disease, it is very difficult to overstate the pain and the misery that came along with this disease. Right, it was a terrible, painful disease that often resulted in death, or if it didn't kill, it caused lifelong injury. So some symptoms that would come along with this are not just a high fever, but also these body-wide rashes that you could contract that would ultimately become scarring sores. It would cause intense fatigue and vomiting and a number of other symptoms, and it could kill. In fact, it did cause about 35% mortality, 30 to 35% mortality historically. So it was very, very contagious as well, right? So this is a very problematic disease. This is the virus here, variola major. This is what it looks like. So it was around, actually, I'm not entirely sure when it was initially figured out. It's a very old practice. People realized very early on that you could infect somebody with material from smallpox, from a smallpox infection, like a pustulous sore, and if you did that in a very specific location, you would induce a minor infection, right? You would do a minor exposure that would induce a minor infection that would result in minor scarring and a very, very strong immune response. And it also carried with it a lower rate of mortality, right? So this was a process called variolation. And so people would do variolation of smallpox. It would look a bit like this. First, you would extract a bit of fresh pus. This is very gross, but you would do this. You'd extract a little bit of fresh pus from somebody's smallpox sore in a live patient. And it was really important that you looked for a case that was mild to do this from, right? You'd want to go and find somebody with a very mild case, and then take the fresh pus out of a sore that they developed. Okay. Now we know today, they didn't know this at the time, but we know today that this material is infectious, right? That it carries that virus, and so that's really what we're doing. We are cultivating material that contains the virus. You would then allow it to dry and then grind it into a fine powder. Okay. Now, then from there, you would administer the powder in a few different ways. One way that has been documented is blowing into the nose, but a much more common and more widely used method was scratching it onto the skin. And then you would wait for the patient to develop symptoms of their infection, and this would be, by and large, a more minor case of smallpox, right? That they would develop and then swiftly overcome in most cases. I want to say in most cases because again, this is variolation. This is the progenitor concept to vaccination. Vaccination today is so much safer, and most vaccinations today don't use live virus material or unbroken, undamaged viruses. This variolation did, and so it carried a higher risk associated with that, among other things. And we learned a few different important things from this process. First, we learned that that dose was very important, right? The viral load that you are exposed to initially is very important for determining how severe your infection is going to be, right? That you wanted to get a material from a case that was very, very minor. The scabs and the pus and things like that you would cultivate had to be from patients that were actively fighting and doing very well for the disease, not just because it had antibodies and things like that, but also because it was a mild case where viral load was probably pretty low to begin with. But importantly, we quickly learned that the where and how the virus, the powder was administered mattered a lot. We quickly learned that if it was encountered through the respiratory system, it tended to be much worse of an infection, specifically like a body-wide infection, than if it was scratched onto the skin. So variolation that was done in this picture, where you have this probably pretty unhappy child that is getting this variolation scratch in this local area, what would happen is this case would develop into an infection that would probably encircle part of the arm. You get a lot of rashes around the arm itself, and then they would heal over time because your body would fight off the virus. This is because the port of entry through the skin is much less effective for smallpox than for the respiratory system. The disease spreads best through the respiratory system, right? And so we quickly learned that it was best to avoid this port of entry, which is why blowing the material through the nose quickly fell out of favor, right? And most people began to do the skin contact method instead. Where this infection was a slower infection, it was much more localized, and it was much easier to overcome with the body's natural immune system, right? So that is all for this lecture. Please remember, as always, to read the book chapters. This corresponds to chapters 13 and 14. Make sure to read that. And I will, there's one more to go. I will record that one very soon and have that up for you. Thank you very much. I'll see you in class.